10 after.
Last month. And in this session, we have Dr. Akshay Anand. He is an associate professor, Department of Neurology, Postgraduate Institute of Medical Education Research, Chandigarh, Vijayawada, and he will talk to you on retina, stem cells and retina.
Thank you very much, Dr. Anand, and everybody in the organizing committee, in particular the faculty. I could not join you all from the beginning, but from whatever I could gather in the last 2-3 days, it was very interesting. And in particular, I enjoy talking to and learning from students and it is also challenging because talking to experts all the time is very easy, but speaking at the level of the students is more challenging. And also the learning process also is very different. The dimension is absolutely different. So I brought a lot of data which was very, very complex, so to say, for a student who's come to learn and be provoked into neuroscience. So I cut off all those slides and will present very, very preliminary data in giving you attempting to give you an idea of how a study could be designed, how a model could be developed, and then how could we use it for addressing our local problems, whether they are communicable diseases of the brain or non-communicable diseases of the brain. And in that attempt, I would encourage you to be more interactive and stop me in the middle if something is not very clear, and that will probably change the overall thrust of the talk as well, because I'm not very good at speaking to students at this level. As I said, the specialist meetings are something which is more something very more comfortable to talk to. But as I said, I look forward to this, and I've been enjoying the last 2 years. So this is a study which is going to change the focus of the real focus on the brain to a little bit of a retina. That means it is also a part of the brain. It's an ectodermally derived part of the brain, developmentally speaking. And our lab has been trying to address this devastating disease called the AMD, age-related macular degeneration. It's a degenerative disorder of the eye in which the central vision is lost completely. And this was until very recently thought of as a disease of the West. And a lot of people argue that it is probably because in the developing world that the diagnostic capabilities were not good enough, or the aging in general, the lifespan was not as extended as it is now with the development and stuff like that. So more and more cases of AMD now coming to be noticed in India, in China, I don't know for Sri Lanka. So it's certainly a very important disease, and those are the prevalence and the incidence figures. And that tells you how much importance has the disease attracted in terms of funding. And there are actually 2 types of AMD. One is the dry form, which is characterized by geographic atrophy. One is the wet form, in which there is neovascularization, and which is bad, which leads to a lot of blood being, as you can imagine, spilled into the retina, and there is spatio-temporal occlusion of vision, and the central vision is lost. And it is also one of the considered to be the legal blindness, as they term it, in terms of compensation and in terms of being handicapped. So this is how a typical histology of AMD presents itself. And you can look at these drusen-like particles, which are one of the characteristic features of AMD. This is the human fluorescein angiography of a fluorescent-looking drusen particles, and this is the histology. And you can appreciate these amyloid beta rich C5a immunoglobulin-rich drusen particles. Do I have a better?
Yeah.
Actually, I don't have much space to move. I like to move and go into the people and talk, but I think maybe I can move around a little bit. So these are the drusen particles, and they are sitting in the eye, and as the age advances, this begins to coalesce with each other, and there is spatio-temporal occlusion of these particles, and there is a vision loss. And this is the RPE, the retinal pigment epithelium, which is the main culprit behind the disease. And this is where if there is disruption, this is one of the first few features that prompts ophthalmologists to write the diagnosis that this person is suffering from AMD. We were very lucky to come up with the first animal model of AMD, which could mimic the features closer than the other models that were proposed earlier. So we could actually see these drusen-like particles in these MCP1 and CCR2 deficient mice, and they were very much like the human presentation. They were rich in C5a, IgG, immune deposits. Until at that time in 2002, there were no reports of CFS gene and CCL2 gene being associated with AMD. And most of what was believed then was an immune reaction, an uncontrolled immune reaction, inflammatory reaction, and absence of scavenging ability of the macrophages because of the CCR2 deficient mice, because they could not recruit the macrophages to the area of retina and scavenge the deposits constitutively. And that is why the drusen began to develop and accumulate, causing AMD. These are the electron micrograph. We can appreciate the disruption of the RP, retinal pigment epithelium. So, in terms of therapy, there is nothing much that has come up successfully, and therefore there is not much hope for AMD patients as of now. I have not added one particular molecule. If someone points out, I will talk about it if you ask me, and that's a prompt for you. The therapies that they exist today around the world are pretty much thermal laser photocoagulation. You use the laser to ablate and to cauterize the new vessels being formed in the retina in the rect form, which is the most devastating one. And that's the only one that is there as of now, but it also has very limited efficacy. Photodynamic therapy is also used between patients. It depends on what candidates are chosen and what are the types of patients, whether it is wet form, whether it is dry form, or what is the degree of burden of the disease, what is the severity of the disease, and stuff like that. So where do we look for? Which way do we go? We have a problem and the existing therapies appear not to work, and there is nothing like involvement of a chemical or a synthetic biologist, such as one of the people who just talked, two talks before me. I think a really very electrifying talk, very nice. There is no synthetic molecule that could be thought of as an intervention into this disease, because the biology and the underlying mechanism as we know of it doesn't seem to attract such a intervention at this time, at the level of knowledge that we have now. So people begin to think a lot about stem cells. We have a sort of a hype about stem cells. Everyone is talking about stem cells in liver, kidney, eye, brain, and wherever. So of course, similarly in retina too, there have been a lot of literature coming up, and people trying to test stem cells in vivo, in vitro, and all sorts of studies have come up. And I will briefly review that and try to give you what data we have in terms of using stem cells in some of the animal models. And the animal model that we used also will come to that. But before we jump to that slide, what is known in our understanding of the application of stem cells in retina is this. People have shown in vitro that the mesenchymal stem cells derived from the bone marrow and from cord blood and from embryonic origin can actually differentiate into different layers of the retina. I'll show you very nice and even though it is complex picture of the retina, how there are 7 or 8 layers of retina, and all of them have a specified function, and so we need to repair each of them depending on what disease we are addressing. In this case, it is RPE. If you remember, I showed the pigmented layer, retinal pigment epithelium. So people have been able to even regenerate RPE, retinal pigment epithelium, from all these sources that I just named. And the way to do that is to know what cells you are handling and what markers they are actually expressing, so you can trace them as the cells from an undifferentiated stage goes to the differentiated stage, and does it form any of those 8 layers that I just talked to you about. So those are, people have looked at the differentiation capacity using different markers. As I said, in this case it is NSE, neuron specific enolase, and is one of the markers for neurons. And people have also shown that the human bone marrow derived mesenchymal stem cells can also differentiate into photoreceptors and other layers of the retina. And this is just to show that nestin is a very important molecule. It's a very important marker which specifies the neuronal lineage. It's a marker for neural stem cells. And as the differentiation begins to proceed, the expression of the nestin is very well known to recede, while the TRK, the tyrosine kinase expression begins to increase. Its expression goes up, so which is a marker for differentiation. So there are many markers, hundreds of them, and we can choose them and help them to help the known whatever knowledge we have on these markers to trace how the stem cells are actually moving from an undifferentiated state to a differentiated state. So this is just to show that. And there are for the different layers, and maybe I should go to that particular slide first and then go back, because I should have actually put the slide before, but this is just to appreciate the different layers of the retina. So we have a choroid, which is a vascular tissue, and this is the RP, which is involved in AMD. And then you have the photoreceptors and outer plexiform layer, bipolar. So there are 8 layers, and this is where the vitreous is. This is how the light comes in. So this is the first layer of tissue or the cells which are confronted by the light, and this is the most outer layer, the choroid. And this is the one, as I just said, this is the section, this is the schematic. And therefore, all the models, including ours, is centered around this particular layer, the RPE. So for different layers and different types of cells that I just showed you in these layers, there are different markers, and people have studied them, expressed them, and validated them. And so they've done all sorts of studies to show these bone marrow derived stem cells can actually differentiate into different layers, and those markers are for different layers. So calminin is for example, bipolar cells, PKC is for another layer. So that like that they have shown that this differentiation does take place. But what is lacking in a stem cell field, particularly retina, is dose-to-dose comparisons. What is also lacking is the types of stem cells, head-to-head comparison between the types of stem cells which are being experimented as to which is better potential, better efficacy, toxicity or whatever. So those sort of studies are lacking, even though a lot of ground has been covered in terms of finding out the mechanics of trafficking of stem cells as to what signals they use, what signal transduction pathways they use, and what markers they express. But the very elementary thing which is most important in terms of our clinical